Hello, everyone, and welcome to today's webinar, Expanding Biobanking Size and Scope to Advance Big Data Discovery, presented by Dr. Jennifer Coe. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Brooks Life Sciences. To learn more about our sponsor, please visit brookslifesciences.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, just simply type them into the Ask a Question box located on the far left of your screen and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present to you today's speaker, Dr. Jennifer Coe, Associate Staff in Dermatopathology, Cleveland Clinic Medical Director, Central Biorepository, Cleveland Clinic. Dr. Coe is the Medical Director of the Cleveland Clinic Central Biorepository, housed within Pathology and Laboratory Medicine at the Cleveland Clinic in Cleveland, Ohio. She is also a dermatopathologist with an interest in melanoma, Merkel cell carcinoma, and tumor immunology. She completed her BS-MD in the 6-year program at Northeast Ohio Medical University. She then completed an internship at Cleveland Clinic in Internal Medicine and a PhD in Immunology at Case Western Reserve University. She is boarded in Anatomic and Clinical Pathology and Dermatopathology and received all of her training at the Cleveland Clinic. Dr. Coe, you may now begin your presentation.
Good morning, everyone. Thank you for the introduction. Today, we'll be talking about expanding biobank size and scope with an emphasis on our experience here at the Cleveland Clinic to advance big data discovery. So when we talk about big data, what we mean by big data is extremely large data sets. And a lot of the data sets are too large and complex for traditional data processing applications. So they need to be analyzed computationally in order to reveal patterns, trends, and associations. So although big data can be thought about in multiple different contexts, when we're talking about science, we mostly refer to big data as the data surrounding genomics. So DNA sequencing, RNA sequencing, and expression arrays, and the analysis of that. And how do these predict disease course? So what is the prognostic information that comes from analyzing genomic and environmental data? And then what is the predictiveness of this data in far as how will each patient respond to treatment? So big data really helps us get more towards personalized medicine too, because when we look at somebody's genome or their expression, for instance, in a tumor microenvironment or in other tissues or fluids, we can predict how that particular person might respond to a treatment as compared to before, when we would treat everybody the same based on broad disease classifications. So at the Cleveland Clinic, I'm going to talk about things that we're doing to be able to contribute to big data. Cleveland Clinic is probably the first or second largest hospital system in the United States as far as volume of patients, volume of surgeries, complexity of disease. And so we really do want to contribute as many samples as we can to this type of endeavor. So we'll discuss kind of the current/historical state of biobanking at the Cleveland Clinic. It is a process in evolution, currently rapid evolution as we speak, and that's why current and historical are together. We'll talk a little bit about the Central Biorepository in particular, which is housed in Pathology and Laboratory Medicine, and kind of the future directions of the biobank here. So historically and currently, there were several, quote, biobanks within the Cleveland Clinic enterprise and the associated Learner Research Institute. These biobanks could be surgeon-owned, and really it could just be a surgeon who bought a -80 freezer and put their samples, residual samples, if you will, in there, or it could be PhD investigators in learner research. That's the bulk of them, or MD PhDs or MDs doing research. Most of the banks are in learner research, and most of the specimens are tracked on Excel spreadsheets. Some of them have paper, and we've recently done a questionnaire to get it more granular with this data, and some actually didn't even have an answer for that question of how they're tracking. There's really no universal lab information system which connects all the patient-derived samples in the Cleveland Clinic. There's no universal standard operating procedures or freezer monitoring system that connects all the banks in the enterprise. Patient-derived tissue almost always comes through pathology, which is where the central biorepository is, by necessity because of it coming from the operating rooms and needing to be grossed by pathology to select tissue that's appropriate for research. But it's not necessarily tracked after it goes to a principal investigator. And rarely tissues come from outside institutions to the labs, but that's more rare. And patient-derived fluids, mostly we're talking about blood, can come from the Central Biorepository in Pathology. It can come from Learner Research Institute. A lot of these labs have their own phlebotomists, or outside institutions. The Central Biorepository is housed within Pathology and Laboratory Medicine. That's RT-PLMI. Our chair is Dr. Brian Rubin. We have an executive director. And then our pathology is split in between anatomic pathology and clinical pathology. And really the biobank has historically just been in anatomic pathology because most of what we were collecting historically was tissue. And I'm the medical director. I have a technical director, Brad Skilton. We have a rotating PA, pathology assistant, who is assigned on a weekly basis to be the biorepository PA. That's off to the right. They basically need to put all the specimens marked for procurement first on their priority list. We look at a nightly list. We look at the OR schedule the afternoon before, and we print out the list. We give it to the PA. The PA has to know what to expect, what they're responsible for collecting, and we also note the cold ischemia time for these samples between removal from the patient and processing. So the PA works with the tissue procurement techs. We have 4 right now, and the laboratory coordinator to make sure that we collect all the specimens, as many as we can that are targeted for collection in a timely fashion. And then we have a lab assistant as well. So our activities right now are really divided into 3 broad categories. We support clinical trials in the Taussig Cancer Institute. That's all the way to the left. What that is entailed is block and slide, tissue paraffin block and H&E glass slide retrieval from the archives, block selection in conjunction with the various pathologists, and then cutting and sending diagnostic material for patients who are entering onto clinical trials. That used to be run through histology, but the turnaround time was not optimal. We have a 2-day turnaround time for that. We also support IRB-specific research protocols. Most of the PIs are in the Learner Research Institute. Many of the samples that we collect come out of the OR and get caught and released, meaning that we procure and then we release to the PI, and we're not really tracked after it's gone to that PI. We just note that it went to that PI. And under here in green, it lists the number of specimens that were collected in each of these categories in 2017. We're in the process of analyzing data for 2018. And then we have our non-specific tissue and fluid collections. So for that, we have global IRBs. The one on the left is for de-identified, non-consented specimens, and the one to the right is for consented specimens. And we work with the Taussig Cancer Institute primarily to, we do have the ability to consent patients as needed, but that doesn't happen very often when it's directed by us. Usually that has to be directed by clinicians who want to piggyback onto this broad IRB with amendments. So those are either de-identified or consented specimens, and they are usually banked for non-specific future use. And that could be mostly what we target for that are cancer and normal adjacent tissues, inflammatory bowel disease, and epilepsy, because that's like the areas that we've been asked to collect on. Now, the Central Biorepository has space in surgical pathology for tissue procurement. We also have underneath that, one floor down, our wet lab. We have a processing lab, 1,800 square feet. We have 300 square feet for block and slide storage, and then 780 for -80 freezers. Right now we have 14 -80 freezers, and we also keep down there things that are frozen for diagnostic use, such as direct immunofluorescence studies, which are done on frozen tissue and are required to be kept by CAP for 10 years. And we monitor all of our equipment within the biobank in pathology and in all of our freezers in pathology and laboratory medicine with Reese electronic monitoring. LRI monitoring is variable and PI dependent. And so that's kind of the background of the biobanking here. Where are we going? Well, it really took biobanking to come to the attention of our CEO, who at the time was Dr. Toby Cosgrove, for this to start moving in a more futuristic direction towards big data. So Joe Biden, who was the vice president at the time, came and visited Dr. Cosgrove to discuss the Moonshot Initiative. At the time, he, knowing that the Cleveland Clinic is such a large volume institution, asked what kind of clinical translational specimens that we have that could contribute to big data discovery. And at the time, what we realized is there's really no way to know throughout the enterprise what we currently are housing that was patient-derived, and to quickly attach data to that. And so, we weren't in a position that we wanted to be to contribute to big data. And so because of that, Dr. Cosgrove asked to bring together a steering committee for a broad enterprise-wide biobank project. And so it really brought together the Lerner Research Institute, our chair, Serpil Erdrum, Pathology and Laboratory Medicine, myself as the medical director of the Biorepository, and the Center for Clinical Genomics, who is run by Wilson Tang. And we came up with a vision that we presented to Dr. Cosgrove, and that's kind of what I'm going to be drilling down on throughout the rest of this talk. But there's several components of the biobank vision. One is recruitment and enrollment of patients. That requires a lot of collaboration with the IRB and mindfulness of the common role and changes that may be coming down the pipeline. EPIC is the electronic medical record that we use, and how do we integrate patient consent status, patient consenting into EPIC, research orders into EPIC, and data. How do we extract data from EPIC that we can attach to biobank specimens? So IT informatics is huge for that aspect. Sample processing by standard procedures throughout pathology, as well as the Lerner Research Institute. And then a biorepository. We wanted to look for off-site storage. And really it was important to Dr. Cosgrove that we get a strategic partner to do that, somebody with more experience in the field to get us up to speed more quickly, which is how we came to work with Brooks. And we are running out of space in our research institute, so we decided that off-site storage with that partner would be most advantageous. And then governance for access. But this requires the engagement of the researchers, the institutes, the clinicians, and the patients, and the community. And so the goals obviously are many. Scientifically, we want to get more access to both materials and data in a common format and reduce redundancy. We want to become eligible for more future grants based on the organization and tracking of these specimens. We want to minimize physical disappearance of materials. So, for example, when PIs leave and go to other institutions, or lead technicians leave, or graduate students, et cetera, increase the security of the patients. So, minimize risk of noncompliance. We want well-documented material, controlled access, increase access to that material, reduce operating costs, increase efficiency, and basically help caregivers work to the top of their license. So this is a lot of goals here. We thought that we would need to phase these goals into several phases. Phase one really is in the first year. We need a cold storage policy, and part of that would be to make sure that people are tracking their samples in a universal tracking system, and that they are cleaning out their freezers, and that they are perhaps going through the central biobank. Standard operating procedures implemented across, that goes with the cold storage policy. An enterprise data tracking system. So one of the biggest things I think we all recognize is that samples are never to be housed really in one location per se, but we need them to be virtually housed in one location electronically to allow us to really understand what we're doing as an enterprise. And we wanted to establish a Cleveland location for the off-storage biobank site. Phase two would be CAP accreditation for the biobank, biospecimen evaluation and registration. So what do we currently have, what's worth keeping, what's not worth keeping, and get them registered in the universal list system. And then relocation of those registered freezers. And then phase 2 to 3 would be having the established off-site storage, reduce freezer footprint on site, sample collection with standard universal SOPs, streamline workflow, managed and searchable samples, and then that would allow for more industry, pharma partners, and quite frankly, other academic institutions. We've already started to establish some of these collaborations with other institutions, and we're very excited about that. So getting back to the vision, I want to break it down into the different components and talk more specifically about each one. So recruitment and enrollment is really the start of where all of this starts. And the first thing that we worked on was a universal opt-in consent. We had it approved by the IRB. We thought that it would be wise to first target all the patients going to surgery. Pretty much if you're having surgery, you have significant disease that can be classified, whether it's cancer, whether it's cardiovascular disease, valvular disease, epilepsy, there's several orthopedic problems. So we wanted to target all patients going to surgery. We have around 40,000 surgeries per year at the Cleveland Clinic. So we reached out to surgery and anesthesia and established multiple points where patients can be approached for consent. So we really want to get away from the practice of hiring people whose sole role is to find and consent patients, because it's just not efficient. When we looked at, some of these research coordinators who consent patients, they could spend days just scrubbing all the different physicians' schedules in Epic to get to the right ones that they want to consent. And so, we thought this would be a good way to start. So surgeons can approach patients in their clinics. Now, we know that all surgeons are not equally dedicated to research. So for the ones who are, that would probably stop there. Now, the ones who aren't, they might get through the cracks. But then we've engaged anesthesia, and we have PACE and impact clinics where patients get cleared for pre-op surgery, cleared from a cardiovascular and a pulmonary standpoint. So anesthesia will have an opportunity to consent the patients then. Eventually, we see it going to MyChart to consent patients. So then, right now, as it stands, in order to get an opt-in consent approved by the IRB, Cleveland Clinic is a little bit conservative in this way, an opt-in consent could not allow for extra blood tubes. It could only allow for standard of care residual blood and fluid and standard of care tissue, which obviously we had been doing for a long time in pathology. And currently in pathology, and this is a problem that we're going to need to address that I can talk a little bit more about later, currently there's a 3-day hold on all specimens in clinical pathology, so CBCs, CMPs, and so forth, in case that they need to go back to those tubes and do add-on labs. But blood gases are immediately discarded. With blood gases, depending on how many there are throughout the course of the surgery, they come in a syringe, and you get about 1 ml, and then 200 microliters are used for the test, and then 800 microliters are disposed of per draw. So that mostly what you can do is genomic studies. You can't really do a lot with live blood cells. But DNA studies can also be done on samples that have sat for 3 days. RNA studies and cell viability studies are not possible, though. So we plan to collect and pool all blood gases. We've piloted this, and we're in the process of analyzing the data. We will get about 1 to 4 mLs per patient who has blood gases done. And like I said, this will need to be modified in time. We do have a BioBank website that we're building. It will go under myclevelandclinic.org. This will be an access point for both patients and investigators. And it was important to our IRB when they approved the universal opt-in consent that we have a contact listed on the IRB, frequently asked questions, and a contact where patients could email or call with specific questions. So the second piece of the vision is really Epic orders, our electronic medical record, the data in the electronic medical record, and IT informatics issues. And so Epic is in the process of expanding its research module to allow for research blood draw order placements and to assist with targeted patient searches and reporting for research. And so we have people who are working with Epic to assist with that. Universal specimen storage and tracking lists. So the Pathology and Laboratory Medicine Central Biorepository have been using a specimen tracking system that was supported by a company that went out of business. And so we had already purchased LabVantage with several licenses for our own use. At the same time, the need was identified for an enterprise-wide biobank effort, and we thought that LabVantage would fit the bill for that. And so we're in the process now of, we've implemented LabVantage for our own new specimen registrations. Actually started in January of this year, so 2018. We've been registering samples that we collect and that we store in LabVantage, and now we are onboarding the other labs. And so that has been a very interesting process. You know, labs have different needs and different utilizations. We have a team that helps onboard, but I think in these early phases, we're looking to categorize labs into categories, and those categories are still being defined based on their needs, and then doing the student becomes the teacher to other labs who can assist, because it's just, you know, it takes a lot of people to onboard. It's not a very simple system, but because it's not simple, it will be able to fulfill all of our needs for a long period of time. So really, big picture with IT informatics is that we use the Teradata. So Teradata is a system that Cleveland Clinic had bought separately to manage all of its data. And it's a big kind of workhorse engine to do that. So our vision is that Teradata will be a space where the biobank will have its own data lab within the Teradata to interact LabVantage with the other systems. So the Epic electronic medical record, SunQuest, which is the tracking and reporting system for clinical pathology, Cerner CoPath, which is the tracking and reporting system for anatomic pathology, REDCap for different labs using that to track patients, etc. And we have different data warehouses. For instance, in the Taussig Cancer Institute, there's a cancer data registry, and we would like to be able to interact with that. So as an example, you know, if we have an investigator looking for patients with breast cancer of a specific type, we may be able to go into LabVantage and say, we have this many breast cancers that have been collected, and then that we know that we have material on. And then LabVantage would then send those list of MRNs to CoPath to get all of the staging information and the molecular data associated with them. Because, for instance, maybe the investigator only wants ER, PR, HER2 negative breast cancers. So we would reach out to CoPath, filter the list based on who fits that criteria, but then maybe they want to know how many of those patients were premenopausal. So then we would be able to go into the tumor data registry and look at menopausal status and other pieces of information. Not that we would store or duplicate that information, but we would really be able to have a complete data set attached to each specimen that we give that fits the needs of the PIs. So that's kind of future state and hopes. So when we got LabVantage, what we actually had to do, and really LabVantage implementation did not take off until our institute realized that we need people to support it. And when we've talked to the other hospitals who use it, they're using between a team of 5 and 7 people just to support LabVantage. And it makes sense because it's kind of like CoPath or any other institute-wide system. So we were able to hire a lead systems analyst who's also a computer programmer, 2 data analysts who have been very instrumental in going out and assessing the labs that we're onboarding and onboarding the other labs, and then technical analysts, which includes a lot of our computer programmers. And this has been very, very helpful. So getting back to the vision and the IT standpoint with LabVantage, like I said, we went live January 1 of this year. LabVantage does currently communicate with Epic in a limited fashion. So what that means is when we have a specimen that comes in, we scan the requisition on that specimen, and it uploads the pieces of basic demographic information and auto-populates those fields in LabVantage. So that includes like the name, the date of birth, the medical record number, the gender, the ethnicity, I believe. So that stuff auto-populates, which is great because it reduces the room for human error and it increases efficiency. CCCB is, like I said, onboarding other institutions and principal investigators gradually, and our analysts are helping to do that. And we wanted to communicate with all these other systems. So the next aspect of the vision is to get samples processed by standard procedures based on expert recommendations. So whether you get a sample, you know, from anywhere or any year or time point, you know, it's processed the same way. And we really want Brooks, and Brooks has started to assist us with this because they have a long track record of being the biobank for a lot of different big data projects. So they will assist in creating these policies and procedures, which will be uploaded for everyone to see and will become universal throughout the enterprise. Phase 1 is really verifying and registering our current inventory. In that process, for instance, you develop a screening protocol. So first off, anything that's not labeled gets discarded. And then you start to decide what are your second priorities. So, you know, random checks on specimen integrity from different years, from different places in the freezer, these are the sorts of things that we're looking at. What's worth keeping and what do we actually have? And phase 2 would be moving all low-touch specimens off-site to the off-site location where the building that Brooks will run. And so the low-touch specimens are yet to be defined, but pretty much it's going to be anything that's not a reagent or any sample that's not going to be used immediately, because the turnaround time to retrieve specimens is 24 hours. And we hope that many people choose to work with the enterprise. Although we can't force people to give up their freezers, we hope that they see that they don't really want to carry a pager for 24 hours a day and 7 days a week in case their freezer goes down. Or if they're not currently carrying a pager, they don't want to worry about whether their freezer went down and they didn't know it. And they really are not as good as they think they are at keeping and storing specimens, because biobanking, is a job in and of itself, and it's not necessarily the science that these people are good at. They need more time to be reading the literature, designing experiments, analyzing data, writing papers, etc. And so we may eventually need a cold storage policy with teeth, but we're hoping that everyone chooses, when they see the CAP accreditation and the ease of use, to work with the Central Biorepository and with Brooks. So we're starting to inventory the Cleveland Clinic freezers. We distributed files to the Learner Research Institute department admins. They completed an inventory. We reconciled that inventory with a ledger from facilities engineering of all the freezers in the departments. We're working with additional admins to fill the list. And really, we want to complete this process with the help of Brooks in a walkthrough in the winter of this year and next year. So the off-site storage was important. It's part of the vision. And like I said, that's where we're working with Brooks. I can show you now that we are going to be building a building that essentially sits across the street from the Clinical Pathology Laboratory building and the Learner Research Institute on Innovation Corridor, which has been, something that the leaders of Cleveland as a city want to develop. And it's going to be a 21,000 square foot building, 2 floors, about 10,500 on each floor. It will have another footprint of 8,000 or so expansion area and a parking lot and on Cedar Avenue. So this is what it's going to look like. It's going to house -80 freezers, liquid nitrogen freezers. There's going to be a blood processing area, as well as a blood draw station and a research access center. And we'll get into the research access center a little bit later. So it was important for us to engage the community to get approval for this building to be built. We actually had to purchase a residence home in order to build it. We had to work with churches and so forth. And so it's been important that we express to the community that one of the purposes of the biobank, really a major purpose of the biobank, is to get samples from this community to study and solve problems that they're facing with their health. And so we designed the Research Access Center that will be visible at the entry to the building to really showcase the types of research that are going on in a translated version that the lay public can understand, to give health fairs, to give journal clubs and research talks. We're also partnering with local schools. We have Stokes interns, which come from the local high school and do summer internships in different labs, and with the Cleveland Clinic to learn about future careers in research and in healthcare. We'll be organizing educational tours about biobanking and the building with different religious and civic and school groups. Like I said, we want a blood draw space, so if people want to donate blood, for research, they don't have to, go and pay to park and try and find something buried inside the Cleveland Clinic. They can just pull up and come here. We also want to work with other hospital systems, University Hospitals of Cleveland, which is associated with Case Western, and Metro hospitals are also big hospitals in Lerner, or big hospital systems within Cleveland. And so basically they want to increase the foot traffic between the community and Cleveland in a scientifically enriching way that benefits us and them. And also they want to build some shops around here, so Starbucks, Panera, etc. And here's an example of the Lewis Stokes, some of the Lewis Stokes scholars that we've had. And here just shows that we have a clinical research unit, but that's, again, buried inside the center of the hospital. So we really want the blood processing lab in this biobank facility to work with the clinical research unit. And so the last aspect of the vision really is the governance for access. But what I'd like to stress with that, and we really haven't talked about that as much yet, because we need to get the specimens, guarantee their integrity, classify and be able to report on them first. But what I've noticed, and some of you have probably experienced this as well, is that when you start a biobank, oftentimes people think their eyes are bigger than their stomach. So they think that they want to do all this, and they don't want to share specimens. They're worried about other people taking their specimens, so forth and so on. But there's often more than enough to go around. And if we get to a point where we've got characterized specimen sets that people are arguing over, I think, and that we need to govern for access, I think then we're in the place we want to be. I always say you can't call it a bank without withdrawals. So moving on, kind of when you look at the current to future state, I just sort of want to summarize this. So say that, you have an investigator who requires 300 blood samples from healthy participants to be tested for, a lab value X, for instance, cholesterol. And that the samples must be from men, and they must be between the ages of 40 and 60, and they must have never taken drug Y. Well, in the current state, we would need physicians to identify, recruit, schedule, consent, encourage to show up, and draw blood for 300 men fitting this category. And so e-research would have to identify those. You'd have to send out letters to them inviting them to participate. You'd have to send, it's estimated 5,000 letters to get 3,000 people to participate. Then you have a study coordinator call them, follow up, explain the study, determine interest, deal with many machines, phone calls, etc. Have them come in, and then, sample handling, and processing. So give to the lab, get the results, then pull all the other associated data. And it costs a lot of money. They've estimated $32,000, and the study would take 9 to 12 months just for one project. But in the future state, patients would be consented electronically with the universal consent. Maybe first they would be, approached for universal consent by their surgeon or by, but eventually, future state, they would be approached in MyChart electronically, be able to think it over, read the information in their own home, voluntarily opt in, and then we would be able to search who is opted in and then who meets these requirements. We'd get a notification when those bloods were drawn, and then we would go grab the samples. And really, it would be free, and it would happen a lot more quickly. And the future state really makes big data possible and sustainable. So we need IT and computers to automate IRB-driven, hypothesis-driven specimen searches with matching clinical data. Otherwise, there's too much hands-on work. Universal consent allows us to interrogate genomic data, which is essential for precision medicine. And a universal Cleveland Clinic Biorepository allows us to get the numbers that are needed to really look at the interplay of genomics and different disease and response to treatment. And so at this time, I'd like to open it up for questions that anybody might have or that might have sent in. Okay.
Yes, thank you, Dr. Ko, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located at the far left of your screen, and we'll answer as many of your questions as we have time for. Okay, Dr. Ko, let's start with question number one. What is the most important component of biobanking at the Cleveland Clinic?
So I would say there's two. So the first, I think, is working together. Working together with the different institutions. So as a pathologist, you know, we recognize that the NCI, for instance, it's NCI best practices for biobanks to be housed within pathology. And we are an integral part of deciding what tissue is appropriate for research. And we've long been in the practice of cataloging samples, characterizing samples, the data attached to those samples. But we also need help from clinicians. We need help from the different institutes. We need help from the Research Institute. And there are a lot of, at a place like this, there is so much work to go around that everybody needs to do their part, and there is so much wealth to go around. That the perception that, you know, one person is going to use all of the samples or steal from everybody else is just wrong. And so I think being very transparent and being very, I had to gain a lot of trust, I think, when I came into this position, that that your specimens are going to be available to you. They're not going to be given to anybody else, and so forth. So there are a lot of politics, but I think if everybody is working towards the same cause and really has the best interests at heart, you can overcome that. And then I'd say the second most important piece is IT. We are only as good as our computers, and in this day and age, and the data is so immense that we need the computers not only to really track the data well and be able to tell us what we have and the quality of what we have, but also to get consent of patients, to identify patients that are targeted for collections. I see a time when, in the future, when someone comes to me and a researcher comes to me who's studying basic science prostate cancer and says, I need, blood from this many prostate patients. And then I can sort of go into our files, see how many patients have been diagnosed with prostate cancer. If I need specific attributes, there can be a search on CoPath. But then the automated part comes where every night, once you've got that list of MRNs, you can actually be procuring on a regular basis by feeding from LabVantage to SunQuest each night. Do you have a match? Do you have this patient that we're looking for? Did they come through? Did they have a blood draw? And then you get a report the next day and says, oh, these criteria that you've programmed in to come to me every night is here, and here's where it's located, and now you just go get it. But as long as we're doing everything manually, it's not going to be sustainable. And at a hospital system like this, with reimbursement going down, set by CMS, I think that, we all know that research is going to be the first thing on the chopping block. And so you've got to get more efficient. And the only way to get more efficient is to use computers. Okay, so here we've got a question, which is, what is the financing model for keeping freezers and low-touch inventory off-site? So that's a good question. Right now, the money has been given to, the startup money for this has been given to the Lerner Research Institute as a part of one of their innovations initiatives. The hope is that we will be able to basically recover that cost when investigators write in the cost. So instead of writing in the cost to store themselves, they would write in the cost to store off-site with this off-site location into their grants and into their proposals. But we understand that's going to be a gradual process and that it requires some startup money from the Research Institute. But we hope that it will be worth it, because investigators and their research personnel will be able to work to the top of their licensure and to be working with very high quality specimens. And we also hope that this encourages collaboration. So once patients have signed universal consent, we will be able to be in a place where if you've collected 100 samples and you've used aliquots of those for your study and you no longer require them, or maybe you can open up those samples to be visible and available to other investigators, and the annotation that you've already done with them, even from an experimental standpoint, also becomes available to other investigators, and then they have the opportunity to utilize and look at that data and utilize whatever sample remains in a different way. And it's a win-win situation, really, because it's a collaboration, it's better use of the samples, and I think increased visibility will increase, I like to think of it as a renaissance period, increased trade, and everybody wins. So that's another thing that we're hoping comes from this. All right, I've got another question. How do you handle FFPE samples that are 10 years old or older that CAP has to be destroyed or completely de-identified for laboratory purposes? So my understanding with CAP is that they are required to be kept for 10 years, but whether or not they're destroyed after that or de-identified after that is really up to you. So, we're required to keep things for 10 years on main campus, but we often keep them much, much longer. And I know that some of our satellite hospitals, which were formerly community hospitals, were in the process, or were in the habit of destroying FFPE blocks at the end of 10 years because it was more economical. But there's nothing that says that they had to be destroyed. They were choosing to destroy those. So what we've actually done is tried to recuperate a lot of the ones that our regional hospitals would be destroying, bring them down to main campus, sort through them, and see which ones are very valuable that we want to actually keep in our archives. And de-identified, we de-identify them when they're needed for research, which we would do anyway if there was no consent. So we're not required to de-identify. We're the honest broker because we're within pathology, and so we would de-identify them before sending them out to the investigator. And then how many samples are you currently storing in the biorepository? So that's one of the problems. That number is hard to get at. Each freezer we know can store around 50,000 samples, and there are an estimated 400 freezers around the enterprise. So you can do the math for that. But what we're in the process of figuring out is how full are all those freezers. I'm pretty sure they're not all full. So whatever number you get with that, you can probably divide in half, and that's about how many we have. But the agreement, at least as it stands with Brooks, is that we would be obligated to fill 167 freezers in the off-site storage facility for 10 years. Brooks will be working with other people, like university hospitals, as I mentioned, Metro, and also have other clients besides just Cleveland Clinic. Okay, so what measures are you taking to analyze / ensure integrity of samples for genomic studies, so DNA, RNA degradation over the time that might affect analysis outcome. So we've had summer students that have done projects on this, looking at RNA and DNA integrity. Actually, we sent these samples to Brooks to get their analysis, and perhaps I can give another webinar just reporting some of this more concrete data, looking at the integrity of DNA and RNA in different tissue types over different time points in both cancerous and non-cancerous tissue. And actually what we found, I was surprised because I thought that RNA was basically worthless after a certain amount of time, but that's not necessarily true. We found that there was more variability based on tissue type than there was based on year of banking. And so that's why I think that we really need to go through these specimens more laboriously than just throwing away all specimens before a certain year, because our data would indicate that even 10, 20-year-old frozen samples have good DNA and RNA integrity depending on the tissue type. And most likely, a lot of that is related to pre-processing variables, things that are out of our control, unfortunately. Some of that is warm ischemia time. So when the tissue's in the patient, what's going on with that patient? What's their blood pressure like? Are they acidotic? Have they been clamped off for too long? The blood flow to the tissue before it's removed. Secondly, how those tissues are processed uniformly within pathology. For instance, breast tissue, depending on the size, can be buried in a lot of fat, which can autolyze over time if the tissue isn't inked immediately and sectioned properly, and then tissue grabbed for research and then fixed and paraffin. So just like what you see with FFPE tissue, there's some variability in like tissue and how quickly it's likely to be processed. And that's why we're getting more into tracking all the cold ischemia time, having a dedicated PA that has to prioritize all the research specimens and get them processed immediately. So if we take those variables out, I believe that samples will have good RNA and DNA integrity for many years. So what ethics problems do you anticipate? So we've seen a little bit of this, I think, which gets into so for a long time, I guess I would say that the Cleveland Clinic was like the Wild Wild West. So, you've got a lot of fiefdoms, institutes, and research groups who are doing their own things, and maybe they would be working with different companies, or maybe they wouldn't, and there was separate IRB approval and separate law review and separate MTAs. And I think what we're seeing is that that's not the way that we're going to go. We're going to go to full transparency around the institute and the institution. So part of that is what's everybody doing, and, who is really the governing body? So that gets into that governance piece. Who is the governing body? The Cleveland Clinic owns the samples. It's not the surgeon. At this point, now it may change and become the patient, but at this point, the Cleveland Clinic owns all samples. So it's not the surgeon who removes the samples. It's not the pathologist who signs out the case. And so really, if anybody is going to make some kind of agreement with those samples, it has to be representing the Cleveland Clinic on the whole, not just one person, not just one institute. So that's number one. Number two, we would like to sort of acknowledge and spread the culture that samples that would otherwise be thrown in the trash or incinerated have a high value to discovering and curing disease. And I say that to everybody that I talk to. While you may question the ethics of us using residual samples for research, I question the ethics of us throwing those samples in the garbage when they could be used towards life-saving treatment. And so I think it starts with spreading the culture in your hospital that you accept that this is your ethical obligation to advance research which ultimately helps treat disease. So it is for the patients. Then you kind of spread that ethics to your community and that idea to your community. Look, this is like recycling. This is not being wasteful. This is, and then full disclosure, full disclosure. We would love to consent. Our goal is to consent every patient, Cleveland Clinic patient, and give them a chance. Patients really do want to participate because they know that it could help, not only them, but their offspring, especially people with genetic diseases and so forth and so on. So I think a lot of it is a cultural thing, and we have to be the champions of that. And then, just being honest and saying, look, we have to recoup costs. This gets to sustainability for the work that we're doing for this research, but all the cost gets reinvested into the research and the work related to that research, and it's part of the sustainability. So that's how I would approach that. Now, here I've got a question. Rather than individual manual freezers, have you considered the Brooks automated biostores? So I'm not that familiar with the automated biostores, so maybe I should become so. I'm assuming that means robotics. So before my time, when I was not the medical director, they did have this idea that they wanted to collect blood on everybody who came through the Cleveland Clinic. And to do that, you would need robotics, and it would need to be automated. And at the time, the person before me worked out the cost for all of that, presented it to the executive leadership, and there was no money for it. And so we haven't looked at something at that level yet. I really think the Cleveland Clinic culture is to start with what you're given, show what you can do of it, and then they'll give you money and you grow. So we're kind of starting with what we've been given, and we hope to really show what we can do with it. And I think the best way to do that is target particular disease groups and patients going to surgery, et cetera. Now I have a question. Let me see here. Hold on. I don't know. Okay. In the pharmaceutical industry, we much validate. So installation, qualification, operation, performance are freezers and have a robust collaboration and maintenance program for each to ensure samples are not qualified or compromised. Additionally, the software for control has similar requirements. Do you have this for your repository too? So that's part of the issue, right? We want to collaborate with pharmaceutical industry. We're not currently in a position. We do in the Central Biorepository qualify our instruments in our freezers. We just got the software, which is LabVantage, which will be, qualified, but all the other samples are not in a position to do that, except probably select labs, which either are functioning as biobanks for the NIH, or we have one research lab that actually has CLIA certification in the Lerner Research Institute. And so that's part of the deal, is to harmonize and validate, qualify everything with the help and assistance of Brooks so that we are in a position to collaborate with pharmaceutical industry.
Thank you, Dr. Coe. Do you have any final comments for our audience?
I don't. You can always feel free to email me if you have any other questions or ideas, koj2@clevelandclinic.org or at CCF, so it's for Cleveland Clinic Foundation, but it's letter C, letter F.org. If you have any collaboration ideas and so forth. And that's it.
Thank you again to Dr. Jennifer Coe for her time today and her important research. We would also like to thank LabRoots and our sponsor, Brooks Life Sciences, for underwriting today's educational webcast. Before we go, I'd also like to thank our audience for joining us today and for their interesting questions that we did not have time to answer today, and those submitted during the on-demand period will be addressed by Dr. Coe via the contact information that you provided at the time of registration. Now, this webcast can be viewed on demand through June of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye. Okay.